So the fourth topic, coming from topic 3, is the effects of genes or more broadly genetic variants on development and organism phenotypes. So here the goal would be high throughput measurement of variants in organisms and organoids, not just cells or single cells. So here we would be looking for high throughput assays that use organisms and organoids to test the effects of the variants, different perturbations in different cells, and measuring cell-cell interactions. Definitely an emphasis on rapid and informative phenotypic assays in organisms and animal models, and the data generation expected here would be variant and phenotype atlases. And finally, for number 5, we've got understanding gene-environment interactions. So the goal here would be high throughput approaches to accurately measure E. So context is queen here. You could argue that the single cell level, tissue level, and maybe model organism level, it's actually much easier to measure E. And so here the challenge would be the high throughput assays that would accurately measure the effect of that environmental exposure in a high throughput manner. So the high throughput phenotyping assays here and approaches. For humans, it's a much more difficult problem because we really don't measure E very well at all. So here, NHGRI actually does have a history of really catalyzing our use of electronic health records for electronic phenotyping, and so maybe in the same vein, really catalyzing the use of EHRs, augmented with other data to extract out relevant environmental exposures, maybe merging with other data sets, supplementing with emerging wearable data, biomarkers, even outside databases. So not just individual level environmental exposure data, but community level, socioeconomic status, social determinants of health, outside databases such as air monitors collected by the EPA. And finally, harmonization and standards for human collection of environmental data, very, very important. NHGRI has funded efforts in this area through the Phoenix project. And so we're still in desperate need of this because unlike genome-wide association studies, it's very difficult to combine these data sets across cohorts because there are no standards or harmonization kind of frameworks that have been used to effectively help us combine those data for the powerful studies that we're going to need in humans. So that one is a bit more daunting and multi-year, I would think. So the data generation here expected would be a repository of genotype-phenotype environment relationships in general.